Hello, we are team Desoxyribonucleïnezuur from the TU Delft. We've made a multi-genome visualizer. In this program you can open GFA files and view differences between different genomes. I'll start with opening a file. I will open a 10 tuberculosis file. These are 10 different genomes from tuberculosis bacteria. This is how it looks like. You will see a graph with nodes and edges. Each node stands for a sequence of base pairs, and they are part of these genomes. You can see 10 tuberculosis. All genomes have these sequences, but this is a mutation. These 9 genomes have the C on this location, while this other one has a T. You can move through the graph by panning, just dragging, and you can use the minimap down below to instantly jump to a location or giving your node ID on the left. As you might have noticed, these nodes are not all the same. This is because the length of the sequence matters for the node length. That's why it's easy to see if the difference is just a single base pair or it's actually much larger and more complex. If you want to highlight a genome, say this one, and compare it with this one, you can select them in the highlight tab. You can see both the genomes share this part, but here they separate and each have their own have different base pairs in their sequence. You can follow the paths with the highlight and see this is all part of the TKK040031, and here they join and have the same sequence, and then they differ again. There are other visual highlights you can see if you deselect the genomes. The more blue nodes have more genomes going through them than the less blue. This makes it easy to find clear paths of what is very used. I have a location of that. This is very useful if you have such a big graph as this with a lot of variations. This is another file. This is 328 tuberculosis, but you can clearly see that a lot of the genomes have this sequence, while a few others have a little bit different sequence with also different lengths. Another thing you can do is if you have found an interesting spot which is interesting, so let's say this location, you can bookmark this with the bookmark. Then you can give it a description and a name. So I'll call this interesting, and the description is look at this tomorrow. So tomorrow I'll know I'll have to look at this. Oh, I already have one with this name, so I'll type in the one. And when I go away to another place, say I'm here and I want to go back to the bookmark, I go to my bookmarks, I open this, I double click, and I'm back at the interesting spot I just found. And another feature we have, which is very clear in the tomato file, is we encode SNPs. Those are single nuclear polymorphisms. We can toggle them, which means they will be displayed with a diamond and 2 colors representing the base pair colors. This makes for easy viewing and makes it instantly clear what the differences are. And that's about it, I think. Thanks for watching, and I hope you'll like the program.